In this video, we cover the first analysis steps towards clustering cells and finding marker genes. So the topics I will be talking about is how to create a scRNA object, how to filter genes, how to check the quality of cells, and if there are problems, how to filter out low-quality cells. So what will you learn during this video? Well, after listening to this video, you should know what kind of input files can be used, what is the structure of the 10x genomics matrix file, how to filter out genes, and how to check the quality of cells and filter out bad ones. So let's start with input files. So in Chipster, you can give two types of input files. You can either give 10x genomics files or a digital gene expression matrix or DGE if you have drop-seq data. But let's first have a look at the 10x genomics files. So essentially you have three files and they need to be named like this. So there is the matrix file, barcodes and features, which used to be called genes in the older cell ranger versions. So the main file is the matrix file, where you have the information of the number of UMIs or transcripts for a given gene in a given cell. The matrix is a sparse matrix, which means that only the non-zero entries are stored to make the file smaller. And here we have a beginning of one matrix file. So first there is a header. It's 3 lines, and the third line is the interesting one. So here we first have the number of lines in the genes file. So essentially, this is the number of genes. Then you have the number of barcodes, so essentially the number of cells. And then finally, the number of lines in the matrix file itself. After these 3 lines come the actual information. So here you have, in this column, you have the gene index. So we go to the features file and check which entry corresponds to this number. So here in the features file, we have the identifier for a gene, the gene name, and there would also be a type, so a gene expression in our case. Now, if we continue on this line then, so first we had the index of the gene, then we have the index of the cell. So we go to the barcode file and check there what cell this corresponds to. And then finally, we have the number of UMIs. So this number here tells how many transcripts we detected for this gene in this cell. It's important to remember, so you will actually get 2 folders and you need to take the one which has filtered barcodes rather than the raw barcodes. So filtered essentially means that we only have those cell barcodes that are actually present in your data, because otherwise you end up with this enormous matrix with all the possible barcodes that exist. Okay, so now we have covered these 3 files. When you use them in Chipster, they need to be in a tar package, and then you import that tar package to Chipster, and there should be nothing else in that tar package. Now, alternatively then, as I said, if you have drop-seq data, you probably at this point have a matrix of gene expression values. You can produce that in Chipster, starting from the FASTQ files, or you can also import a ready-made digital gene expression matrix. When you come with these files to Chipster, it's important that you assign the input file correctly. So you need to indicate, is your file this type or that type? And one thing to remember, of course, is that this file can be zipped so there is no need to unzip it. So this is what the window in Chipster looks like. So the tool is called scRNA version 3 Setup and QC. And down here is the input file section. So here, if you have the tar package of 10x files, you need to make sure that it's assigned to this box here and not down here. Otherwise, there are some parameters. So there is a project name. This name is used in the plots. Then if you have several samples you are going to analyze, so for example, control and treatment or whatever, so you are doing integrative analysis, then it's very important at this point that you give a name to your sample. Finally, the third parameter here is about filtering out genes. So there might be genes that are not expressed in any of the cells you have, or that is expressed only in one cell of the thousands of cells you have. So that gene might not be interesting. So here you can set a filter for that, and by default it's 3, which means that the gene has to be expressed in at least 3 cells in order to be kept in the analysis. When you run the tool, you get 2 output files. One of them is R object, and the other one is a PDF file. So here you can see them. So this R object is a special file. It's actually a scRNA object in this case, and it's a special data structure that the scRNA-based tools use for storing different kinds of data. So it has specific slots for different types of data, and in Chipster you use this file as the input for the next analysis tool. So the data, the actual data, and whatever you add to it or the analysis tools add to it is stored here. You cannot view this file in Chipster, but you just give it as input to the next tool. Of course, if you use R, you can always import it in R. But the interesting file for you now at this point is the PDF file, which contains the quality control plots. There we have different terms, so which can be a bit confusing. But so we have the number of expressed genes in a cell. This is called N-feature RNA. There's also the number of transcripts in a cell, N-count RNA. And then there is the percentage of mitochondrial transcripts, %MT. And so these names are now from scRNA version 3 that Chipster uses. scRNA version 2 had slightly different naming for these things. So now let's look at these plots then. So the whole point here is that we are checking the quality of the cells. Remember that in this kind of droplet-based single cell system, what we want to have is we have one cell in one droplet, and that one cell should be a healthy, happy cell. Now, it can happen that there's actually no cell in a droplet. We might still get some signal from this droplet if there is some ambient RNA floating around, but typically the number of genes expressed, so to say, in this, so to say, cell would be very low. And the default threshold that is often used is 200. So here we have violin plots. So every black dot here is a cell. And here we have the number of expressed genes that we detected per cell. So you can see that the majority of cells are sitting here, just a bit below 1000, but then there are some cells which have very low number of expressed genes, and these are the potential empties. Now, on the other hand, it can happen that several cells end up in one droplet. So perhaps 2 cells, so then we have a doublet, or even more cells when we have a multiplet. And this is also a problem because we only want one. So the way to detect this is again to look at the number of genes expressed. And here the default threshold is 2,500. So again, if we go to this plot here, we can see that yes, the majority of cells are here, but then there are some cells which are really high up here. So these are the potential multiplets. Now, it could be that we have one cell in a droplet like we should, but then that cell is broken or dead. The way to recognize that situation is to look at the percentage of mitochondrial transcripts. So how big percentage of all the transcripts in a cell are actually mitochondrial? And so why is this related to the happiness of the cell? Well, if the cell is broken or even dead, uh, the normal transcripts have probably degraded already, while the mitochondria, because it's a separate structure, the mitochondrial transcripts might still be more available or intact, and hence the percentage of the transcripts of mitochondrial origin can be high. So here we have that plot. So now on the y-axis we have the percentage of mitochondrial transcripts, and you can see that for most cells this percentage is like 2.5, but then you have some cells which are really high up here, and it's very likely that these are broken cells or even dead cells. So what we want to filter out in the next stage is these ones here, these ones here, and these ones here. And so this is the tool where you would do it. This tool actually does many other things as well, which we will discuss later, but now let's focus on these three parameters here. So here precisely you say how you want to get rid of the empties. So what is the threshold? How many expressed genes a cell must have in order to be kept? And you also want to get rid of multiplets. So you want to get rid of cells which express more than this many genes. And finally, you want to get rid of the dead cells too. So the default value for the mitochondrial transcript percentage is 5. Now there was another plot that I skipped. So this is basically using the same information, just plotting it differently. So here we have scatter plots. In the first one here, we have the percentage of mitochondrial transcripts versus number of transcripts. Here we want to see, are they actually cells which might have a small number of transcripts in total, and then high percentage of those are mitochondrial, so they are likely to be dead cells. In this other one, we have the number of expressed genes per cell and number of transcripts per cell. So this you would expect that there would be a relatively good correlation. So in other words, if the number of genes expressed and detected increases, then also the number of transcripts or the UMIs should increase. So, so far, both these tools I showed are based on the scRNA package, but I would like to point out that there is also another um, package called scATR, which is very good for quality control. So scATR is a bioconductor package. It has very rich functionality for quality control and visualization of single-cell RNA-seq data. scATR has its own object structure. So the R object that we discussed so far was a scRNA object. Now we have a scATR object, and they are different, but you can give a scRNA object to scATR tools in Chipster because Chipster takes care of the conversion behind the scenes. So there is a tool, Scatter QC in Chipster. It produces many different kinds of quality plots, and there's a separate video explaining how to interpret them.